Okay, this is a voice recording for connections. We're going to try and make as many connections as we can from this guy, my little buddy. This is Watson. He is a wiener dog or dachshund mixed with the terriers, named after Jim Watson. There you go, DNA, double helix. No, he's not. He's actually named after Watson and Sherlock Holmes. But anyway, let's talk about how Watson here can connect to almost every single unit we've discussed this year so far. So, start off. Let's talk about his cells. He is an animal. In the animal kingdom, he is a mammal. Hair, mammary glands, all those things that are synapomorphies of the mammal class in Chordata. And we're going to talk about initially his cells. So here's a generic animal cell. We have multiple organelles. We should talk about the endomembrane system. We should talk about different types of cells and how this is a generic cell. So first things first, eukaryotes, right? Eukaryotic cell here have membrane-bound organelles and a nucleus to hold its genetic material. So here we have a nucleus. You can see the nuclear pores. It's a double membrane nucleus here, so it allows things to transmit in and out selectively. I have a nucleolus where we're going to produce ribosomal RNA and some other ribosome components and RNA components. In the nucleus, then here, all that stringy stuff, that's going to be your chromatin or DNA bound in with protein components, histones. It'll all coil up if we were going to go into mitosis. We have these mitochondria, which we'll talk more about as well, sites of cellular respiration, specifically the Krebs cycle and the electron transport chain, going to make massive amounts of ATP for us. We have the lysosomes that contain those hydrolytic enzymes to break down products inside. The centrioles, where we're going to have mitotic spindles form out of. The Golgi body, which is going to help out with our packaging if we're going to make some proteins to be sent on the cell. We have the rough ER and smooth ER. Smooth ER gets all the ladies because he's smooth. But the rough ER is going to have all these ribosomes on it for protein. And if they're stuck to the rough endoplasmic reticulum, they're going to be transporting those proteins out of the cell. So here's the smooth ER. This is going to be more about carbohydrate production, lipid productions. And then you can see the microtubules and how they not only provide cytoskeleton components, but can be used for flagella if it was a sperm cell. It could be used to produce cilia if it was talking about the trachea of Watson to allow things to move up and down for immune functions. So let's dive in. Let's talk about the endomembrane system as well. So the endomembrane system, a big, huge connection here. Membrane system is composed of the nucleus all the way out to the cell membrane. We exclude the mitochondria because they are not part of the endomembrane system as far as what we're talking about for protein productions that are going to be secreted out of the cell. So if we go or think about his pancreas is going to produce insulin, and insulin is a protein. It's actually a hormone that's going to regulate blood glucose levels by binding to cells around the body that allow glucose then to get into those cells to be used by the mitochondria. So the ribosomes here on the rough ER are going to be the sites for the insulin production. It gets the code from the DNA. The DNA will be then transcribed into mRNA. mRNA will travel out to the ribosome. Ribosome will make the protein. It'll wind through the ER, bud off as a little vesicle, come over here to the Golgi body. The Golgi body will then modify it, add some tags to it, make it a precise 3D structure. It then leaves on the trans side here with another budding vesicle, binds up with the plasma membrane here, the lipid bilayer, and then is exocytotically pushed out of the cell. And if we're talking about insulin, it'll be pushed into the bloodstream. All right, and then it's transported throughout the body. So types of cells that we would find in an animal, we would have skeletal, muscle, and other muscle cells. So I should say muscle cells. We have muscle cells like skeletal muscle, smooth muscle, and cardiac muscle. We have connective cells in the connective tissues. They would be like bone, blood, fat cells. There's a variety of different ones. Blood cells, I said blood cells. Epithelial cells. So these are ones that we would say lining cavities or on the surface. Epithelial Can't write and talk at the same time. I apologize. And then we have nervous tissue as well. And we'll kind of talk about those as we walk through this entire connections video. All right, next up, go into those cells even deeper, and we talked about DNA inside that nucleus. Here's the DNA molecule. You should understand how it's put together and how the bonding looks. So here we have any of the gray is going to be carbon-based things. Red will be hydrogen or oxygen-based things, and then our blues will be nitrogen. So what you're looking at in the center here, these rings are not carbon rings for sugars. They're actually nitrogen bases across the center here. And you can see that this has 2 bonds to it. So this is going to be the nitrogen bases of A to T and not to G to C. Here would be G to C. We could talk about those purines and pyrimidines in a second. So when we're building up DNA, we would have a phosphate, we would have a pentose sugar, and then we would have our nitrogen bases that all point towards the inside of the cell. Those bonds that you see here, those are hydrogen bonds holding them together, which is super important. We don't want a covalent bond. These are covalent bonds here, because with heat we want to be able to separate this, or with an enzyme be able to separate it easily. For example, DNA polymerase would want to be able to get in there and add its new nucleotide. So this piece right here is a nucleotide. We're going to have phosphate sugar bindings of covalent status down the side, as you can see down here. Those are nice and strong. And then, like I said, across the center, the nitrogen bases are going to bind. Nitrogen bases of A goes with T. This is the base pairing rules. And G will go with C. So awesome, teacher. Grant is cool. Yeah. 3 hydrogen bonds with G to C. A with T, it'll just be 2. When we're talking about pyrimidines and purines, purines are going to be 2 rings, 2 nitrogen base rings, or nitrogen rings, not nitrogen base rings. They are nitrogen bases, fused together. So this would be purine. And the purines are going to be A and G, like pure silver. If you know periodic table elements, Ag is silver. Pyrimidine are the smaller rings, so it's just 1 ring, and that would be C and T, or in the case of RNA, our cousin to this nucleotide here, for RNA, it would be a U instead of the T. So pyrimidines, or pyramids, right? If you had a pyramid shape and you accidentally stepped on a pyramid, it would cut you, C-U-T. But these are smaller, and if you cut something up, it would be a smaller, and that's why you only have 1 ring here. So if you look, here we have 2 hydrogen bonds. So 2 hydrogen bonds means it's A and T. So then we can look at it again and figure out, oh, this one's big, so that must be the purine. So right there, that's the A, and then this one would be the T. Here's a big ring right here, so this has got to be the G, and then this one's got to be the C in this scenario. All right, so just being able to reason through some of that stuff. And all this DNA, right, strung out here, double helix, would be, if it was linear, it would be really long, about 3 feet per cell. But that's not how much is in your cell lengthwise, because it gets all coiled up. So the coils coil upon little balls of protein called histones, coil up around those histones, and then each one of those histones makes up what's called a nucleosome. We'll kind of come back to that, because uncoiling these will help out with our ability to transcribe and translate stuff. So those will coil up, and then the coils coil upon the coils, and 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 eventually you get this nice X-shaped structure, which we call a chromosome. So when this chromatin, this complex of DNA and protein, condense, we end up getting a nice solid chromosome structure. All right, continuing on, how does Watson make his proteins? This is all the transcription, translation. Easiest way to remember this, transcription has a C in it, and C comes before translation. C comes before L, so we got to transcribe before we translate. Where do we do our transcription? Transcription all happens in the nucleus. We want to protect that DNA. That DNA is exposed to other environmental things inside the cell. There's free radicals, things that can destroy the DNA. So it's much more advantageous to keep it in the nucleus. So we're going to have RNA polymerase come in and make its mRNA. There's a promoter region up here, Tata box, that the promoter and the RNA work with. And we can talk operons in a little bit. But that RNA polymerase comes in, makes that mRNA, and there's some other details, but we're going to do it simple right now. And that mRNA then leaves and goes out into the nucleus through the nuclear pores we already talked about, finds a ribosome. Ribosome has two subunits, a top subunit and a bottom subunit. This is all made out of rRNA. So you've got mRNA, right, coming out of the nucleus right here. And then we've got rRNA. They bind together. We have a APE siting, right, for translation. And then the tRNAs, which are these things here, are going to bring in your amino acids. The whole goal here is to make a polypeptide chain. So if we go back even into biomolecules, polypeptide, amino acids, we're going to have an amino group, or NH3, and we'll have a carboxylic acid group. So this is your acid side. So that's a C double bonded to an O to an OH, and then amino NH3. You're going to have a central carbon. There'll be another hydrogen up here, and then down here is your R group. And remember, amino acids, there is 20 amino acids that we know of, at least at this point, on our planet and all other areas that we've gone and discovered. And when we make these peptide bonds right here at this P site of the ribosome, we're going to bind together an amino with a carboxyl. And when we do that, we pull out an H and an OH, and those bind together and you make water. So we call this a dehydration synthesis reaction. So when we bind together multiple proteins, or multiple amino acids, we make a polypeptide, and that's what you're seeing coming out here. Each one of these little different geometric shapes is a different amino acid. So there should be 20 if we want to make this diagram very accurate. And then that protein, right, has multiple levels of conformation. So that first one would be the primary, which would be just the order of amino acids. We then can go into secondary, where it would start coiling upon itself, possibly with some hydrogen bonds, making alpha helices, or we can end up getting these folding type structures. We do a coil and it folds across again. That would be a beta-pleated sheet in secondary. Tertiary, we get some more of the hydrogen bonding, some van der Waals forces, some dipole-dipole interactions. But the big thing is we get some sulfhydryl covalent bonds to stabilize that structure. When we talked about insulin earlier, insulin only has up to the third level for protein conformation. A lot of other proteins in our body, and Watson's body, would go all the way up to quaternary structure, meaning that we have multiple polypeptides, like here's one polypeptide. We'd have to make another different one, and they can all join together in a transformer type of deal, join together to make this big, huge protein. So hemoglobin is a 4 polypeptide subunit. So you'd have 1, 2, 3, 4. I'm just going to make an X kind of shaped blobby thing. It's a globular protein. It'll be able to carry oxygen for us throughout our body. So transcription, translation, bam, bam. Remember, translation is going to happen in the cytoplasm. Transcription is going to happen in the nucleus. That was a quick and skinny. Let's keep going. Spliceosomes and epigenetics. So we have mRNA, right? mRNA is going to come out, and it's going to get poly A tail and a end cap on the other side. And spliceosome, what it's going to do, if we were talking about eukaryotic, which Watson is a eukaryote, there's going to be sections of this mRNA that have what are called exons in them and introns. I'm just going to put E and I on here. The exons are going to be the parts that are going to make up our valid gene, and the I is going to be the intron that we need to cut out. So what happens is the spliceosome, which is one of the first things that we've discovered that is a RNA type of molecule. I don't know why I did that small. An RNA type of molecule that has enzymatic properties. It has ribozyme, right? So ribozyme here. It's actually going to do catalytic activity. So the spliceosome will attach to the E, the exon. It'll attach to the other side of the E, kind of folds it up so that the intron kind of comes into a hairpin, and then it's able to cut the intron out. The intron leaves, and it splices together the two exons. So we have this pre-mRNA, and then we'll end up having post-mRNA. And the post-mRNA is what's going to be sent out to the ribosome, so it'll just be free and clear of introns. It'll just be all exons. The problem that we encountered when we were trying to do DNA manipulation is that things like bacteria, prokaryotes, don't have spliceosomes and don't have introns. so we didn't know a lot of this stuff. So we'd insert genes that had introns into the bacteria and couldn't figure out why it was making the proteins it was making, because they're invalid proteins, they were useless. Later we had to figure out that, especially through discoveries with our retroviruses, that we needed reverse transcriptase. So you'd take something like this, an mRNA molecule that has just exons, we use reverse transcriptase, and reverse transcriptase then, I'm not going to be able to fit it all here, reverse transcriptase is going to be able to take this exon-laden molecule and be able to turn it back into DNA. But that DNA doesn't have introns anymore. It doesn't look exactly like the original DNA because it's shorter. It has no introns. So we call that DNA cDNA, or complementary DNA. And that's the stuff that we're going to work with as geneticists, as DNA technology scientists to solve problems that we have in the world. All right, this leads us into some of the epigenetic stuff that we need to understand. Epigenetics, epi, surface, genetics, genes. So we're talking about the surface of genes and how things turn on and turn off different areas that can be transcribed and translated. So let's look into the agouti gene. This one's real good. Scientists were studying identical twins and trying to figure out why identical twins looked different even though they have the exact same DNA fingerprint. So the agouti gene, they found organisms that were fed high amounts of methyl products. So a methyl group is a CH4, right, or CH3 if it's bound into something. But those methyl groups will cause certain areas of the gene to actually not be turned on. So it actually silences, like it says here, the agouti gene. If they were not fed methylation, these are exactly identical twin mice, you not only saw a coat color change, but also an obesity factor increase. So this is brand new type of stuff, but this plays into the part that you are kind of what you eat. Whatever you consume, what you expose your life to, can actually cause different areas of your DNA, if this is our DNA stretch, to be silenced, to be actually wound up tight and not be able to unravel into a nice straight piece to be transcribed and translated. The other thing that we looked into is acetylation. Acetylation works kind of the opposite way, where it would open up some of these sections to be worked on. Right? So methylation, acetylation, all epigenetic control, not only from what you consume, but also what we have found out is what your mother would consume. So feeding a mom methyl diet can actually affect the children and even grandchildren of that original mouse. All right, genotypes and phenotypes. We'll move on, talk about some of the different things that go into understanding how inheritance is passed. Incomplete dominance, right? We're talking here, I'm going to do it as simple as I possibly can. Big R would be red, right? Big R, we'll talk about little r would be white. If this is complete dominance, this big R would be red as well. But in incomplete dominance, we actually get another phenotype from the heterozygote, and we get pink, an intermediate between the two. In contrast to codominance, where big R is still red, little r is still white, big R in this one would be red with white spots. Okay, so that would be codominance. Epistasis control is when you have, if we're talking about one chromosome and another chromosome, we'll say another chromosome downstream here, number 2. This gene right here might code for coat color, and it might say, oh, we need to make the coat of this individual brown. But down here at number 2, this one says whether or not melanin is distributed. Melanin is a key compound that helps with coloration, not only of hair and skin, but also eye color. So this gene right here really affects whether or not number 1 works, and that's epistasis, where one gene affects another gene's outcome. All right? Pleiotropy is where one gene causes multiple phenotypic effects. Right, so we might have something like the gene for sickle cell not only makes your blood cells different shape, but it also ends up making liver damage and joint damage as well. Polygenic is So what I like to kind of think of as the opposite side of pleiotropy, where you have lots of genes, and you only end up having one phenotype because of it. So you talk about that, we kick back to Watson here. We have all these different genes that are going on in dogs, Canis, right? Genus Canis. And a lot of the genes are not a simple complete dominance that you might have been taught originally in your biology career. A lot of these types of things play in. And even sex linked, Thomas Hunt Morgan figuring this out, that there are genes that are specifically on sex chromosomes, for example, the X chromosome. In humans, we have hemophilia on there, we have red-green color blindness on there, we have Duchenne muscular dystrophy. These things are only carried on the X chromosome, so if you're XY, a boy, you're hemizygous. You only get one copy. You don't get any way to mask that allele that might be bad, right? So if it's a hemophilia thing and you get a little H, you got hemophilia. But if you're a female, you could have a big H for normal blood, and you could have little h for hemophilia and be totally fine, because you're heterozygous, you get 2 copies of the X chromosome. So generally, sex-linked disorders are on the X chromosome, not necessarily the Y. There are some Y-linked diseases as well, but this will give you the basis that help you understand things for the AP test. All right, so let's talk about then how gametes are made, right? Meiosis, mitosis. Here, this is showing you another type of mutational piece. So this would be showing you non-disjunction, right? And non-disjunction, disjunction, we end up having a failure of chromosomes to segregate properly. Remember, there's 2 Mendel laws, the law of independent assortment and the law of segregation. Independent assortment says that if it goes light blue, dark blue, light blue, it could also be light blue, dark blue, dark blue, right? It's this chromosome pair does not affect this chromosome pair when they line up on the metaphase plate for meiosis or mitosis, for that matter. And then law of segregation says that when we're talking about homologous pairs, this tetrad here, which isn't super accurate because there'd be some touching here, we get some crossover that increases diversity in the dog and also any other organism that's doing this. One goes one way and the other should go the other way, but you can see here we didn't separate this first pair, and in turn we end up sending 3 of these chromosomes to one cell and only 1 to the other one, resulting in N+1, N+1, N-1, N-1. Non-disjunction disorders can be destructive, right? If it's on a sex chromosome pair, you get things like XXY, Klinefelters, survivable. You can get, on autosomes, you can get trisomy 21, where there's 3 on the 21st chromosome, and that causes Down syndrome, which is survivable. But the bigger the chromosome generally, so if we're talking about chromosomes 1, 2, 3, 4, 5, if we get a trisomy on those, there's going to be severe effects. Survivability, the ability to survive with lack of a chromosome, really the only one that I personally know of, maybe there's others out there, if you know them, let me know, would be monosomy X, right, where we have Turner syndrome, and the female would survive with the sterile. Okay, so those are some examples of some mutations. So back to Watson, right? All this happens in Watson. There's possibilities with Watson. Watson will not be producing gametes because he is neutered. So unfortunately, buddy, that's how it goes. But we still love him. So let's start talking about how he is what he is, right? Go into a little bit of evolution components. So wolf ancestor, here we are, right? And this is a picture of a extant wolf. But wolf ancestor probably looks similar, but not exactly the same as this. All of the dogs that we have domesticated are from a wolf stock, right? Or a fox stock, or a wild dog stock. Regardless, it's some sort of common ancestor that is shared amongst all of these. So how did all this happen? All through artificial selection, right? Selecting for the traits that humans wanted best in their product, right? So we end up having all these different breeds, like the little teeny dachshund dog that Watson is crossed with, all the way up to things like bull mastiffs and very large dogs that are even bigger than modern wolves are. So this is all artificial selection, which is very similar to natural selection, other than it's not the environment that's guiding what traits are more advantageous. It's humans choosing what they want to be more advantageous. All right. So let's lead into some of Watson's arms and legs. So here's a dog's arm and leg. You can see the bones here in the humerus and the femurs, right? The ulna and the radius, the tibia and the fibia, the carpals and tarsals, right? So this would be like our hand. This would be like our feet. Here's our arm, right? We got the humerus, we've got the radius, we've got the ulna, we've got the carpals, we've got the the phalanges and the metatarsals as well. Same thing shown in the dog's front limbs, right? So there's the carpals, all the same bones in the exact same order. We would call this homologous structure, okay? That's huge for your understanding, how key evidence pieces for evolutionary theory. Okay. We're just going to go over a couple of them. This one's showing you how speciation can happen. Allopatric is geographical barrier separation. Sympatric, just totally cross that out. Sympatric would be genetic basis, right? We see a non-disjunction event within the population providing a new species. So let's kind of talk about how this connects into evolution in general. So evolution, right, we're talking about a difference in reproductive success. And that all is based on the fact that there's differences in the population. Nobody is the same. And because of those differences, some individuals are going to have a slight advantage over others in being able to survive and even reproduce. So the ones that have better adaptations for the current environment are going to be able to pass on their genes more than those that can't. It's not necessarily a survival of the fittest. Survival of the fittest, where only the fit survive. It's just a difference, right? So we end up seeing some individuals that are better suited are going to produce more offspring. Those that are less suited, right? That whole scenario. And we can go into more of this with this second video that I'm going to make. But this is the premise of natural selection. When we're talking allopatric speciation, we're talking about somehow the original group gets separated from the original population. And so we end up having two different populations. It can happen with a fracture islanding. It could be mountains. There can be rivers. There's all kinds of different things that can separate a species. We kind of talked about this when we were talking about the newts out in California. You want to look that up. California. And then we talked about genetic changes. I'm having a hard time writing today. California. Genetic things, we were talking about wheat, right, and how there's some genetic non-disjunction that has yielded the wheat that we currently use. Okay. Last thing, bring it back to water, right? Water, H2O, you should understand. This is the key for life. Water is super required, not only in Watson, but all living organisms. It's required for gas exchange and any sort of respiratory apparatus. It's required for moving molecules throughout your body. And it's all due to the fact that water is this molecule that has polarity to it. So it's covalent bond, but it's a polar covalent. So the hydrogens have more of a positive charge, and the oxygen has more negative charge because, as you can see, oxygen is bigger, so it's able to pull more electrons towards it. It's more electronegative. So in turn, if we had two water molecules, what tends to happen is the hydrogens and the oxygens will interact with one another. So we'll end up getting that hydrogen bond that allows for evaporative cooling to work. It allows for surface tensions to be high, allows for the different features that we see as far as states of matter, where liquid is actually more dense than solid, right? And then gas is last for water, which is super unique. You don't really see that in any other substance on the planet. But water is going to be key for life in general. And this is kind of where we're going to pick up next time in the connections part 2.